Today we're going to do a quick review of cellular energetics. This can be a fairly complicated topic, so quick might be a little relative, but here we go. You can't really understand energy and energetics without understanding the laws of thermodynamics. We know there are 4 laws of thermodynamics, and we should know the first and the second. The first law, of course, is that energy and matter, which is connected, cannot be created nor destroyed. So in the example of the diver on the left, as you climb the ladder, you're using chemical energy to build gravitational potential energy. Then you jump, and that potential energy turns into kinetic energy. It hits the water, and that energy is dispersed. It has less potential energy now. And it's all transfers of energy. When the bear eats the fish, it gets chemical energy from the fish, and it uses that energy to heat its own body, as it is a endotherm. So it's all changes in energy. But then the second law of thermodynamics says that the entropy of the universe must always increase. So while an individual organism can eat food and biosynthesize macromolecules, every time that energy and matter is changed, energy is lost, typically in the form of heat, but also waste products and inefficiencies. So you can calculate efficiency by looking at the amount of useful output that's going on. So energy efficiency is going to be the useful output divided by the total output to determine the efficiency percentage by multiplying by 100. So thermodynamics is essential at looking at the changes that occur in energy and is the basis of the rest of cellular energetics. Those energy reactions can be categorized as exergonic or endergonic. This is looking at the total free energy, the Gibbs free energy of a reaction. If the energy of the products is less than the energy of the reactants, energy is released into the system. That is a spontaneous reaction. You're going from high to low energy, so that is favored as possible. But if you're looking at an endergonic reaction, energy must go into the system. That energy has to come from somewhere. So energy is required for an endergonic reaction. The delta G is going to be a positive number because the products are storing more energy than the reactants had, so it's going to be a non-spontaneous reaction. So exergonic reactions are catabolic reactions. The energy is released, therefore the products become energy depleted, they break them down. Whereas anabolic reactions build things up. Endergonic reactions put energy in. You go from less complex to more complex. You take the energy output of an exergonic reaction and you make it the energy input of an endergonic reaction. This is energy coupling. This is essential for life because it's an efficiency that is needed. There will be some loss, obviously. There will be a majority of loss, roughly 90%. But the energy that is maintained will power another process, and that efficiency is essential. So whether it's for transporting proteins or running a motor protein, ATP to ADP, for example, is an exact key marker of an energy coupling reaction. The waste products of one become the inputs of the other. Biological reactions tend to be uphill reactions. They have an energy of activation that must be reached. This, even though reactions should be spontaneous as far as the delta G is concerned, prevents spontaneous reactions from occurring, which is good because that means that we have control. The reactants must be destabilized into a transition state by applying energy in order for the molecules to rearrange into the products and for the reaction to progress. So if you look at sucrase, for example, if you have sucrose, the disaccharide sugar, and you put water in, it should break down because it's going to a simpler molecule. The delta G is less than 0. But it doesn't. It takes a long time. It could take years if you just put the jar in the sun or something. But if you add the enzyme sucrase, it's done in seconds. So even though the reaction should be spontaneous, it's not. If you add an enzyme, the E of A is going to be lower, so it's going to be much more likely to reach the enzymatic reaction. And then the delta G is not going to be changed because the reactants and the products are no different. You have just made it so that you spend less energy for the reaction to occur. Now, enzymes are absolutely essential because they make reactions easier, like we just said, the E of A. So it's not that they change what the reaction is, It just means that it takes less energy for the reaction to occur. How you do this, the substrate of the enzyme, or whatever the enzyme shape matches, will bind to the active site. The enzyme will then use energy or pressure or a variety of things to cause the enzyme to reach that transition state in a more simple way, allowing the molecules to reassemble into the product. So that process, the substrates are converted to products, the products are then released, and then the active site of the enzyme is open again to allow this to occur. We used to think that it was a lock and key, that it was an exact match to the active site which allowed these things to occur because shape is equal to function, but that didn't explain how tightly the enzymes hold on to these substrates. So if you look at the blue example on the right, the shape, the active site, is sort of closed around the substrate in that enzyme substrate complex. It's called the induced fit. So long as the match is close enough, the enzyme will match and will hold on and will complete the process. So this is why some reactions are easier to occur, especially when you look at issues of competition. The function of enzymes are completely connected to the evolutionary history of that enzyme. So they have optimization points for any of those issues of function, especially the factors that could denature. Taq polymerase, which is a polymerase extracted from Thermus aquaticus, a hot spring archaean, is a great example of this. Because it evolved in a hot spring environment and an archaean that is heat-loving, it functions at a significantly higher temperature than most human enzymes. We do see that there is a gradual rise up in the beginning, so as the enzyme gets warmer and warmer, collisions increase and more reactions occur. But once you go beyond that optimum temperature, it begins to drop very rapidly as the enzyme denatures. pH, we see that there is a too little and a too high option. It's basically Goldilocks. It's going to function at a peak pH, and that is again connected to evolutionary history. So a stomach enzyme like pepsin is going to function at a much, much lower pH than something like trypsin that functions in your intestines. Another factor that controls enzyme activity is inhibition. Since the active site is an exact match for the substrate, well, close enough for induced fit, if something were to go into the active site and block it, that would become a competitive inhibitor and prevent the enzyme's function. If something were to bind to a secondary site called the allosteric site, that will shift the bonds of the enzyme enough that the shape of the active site no longer matches the substrate and will again inhibit the action of the enzyme. If you move beyond the tertiary structure and you start looking at quaternary structures, we have a broader group called allosteric regulation. There are active binding sites and there are allosteric binding sites. But when something is in a quaternary form, the binding to one subunit, one tertiary piece, will affect the binding of the others. This could inhibit, this could activate, as we see here. Activators cause all of the open sites to balance. Inhibitors cause all of the active sites to be inactive. Now, those are only binding to the allosteric, therefore it's allosteric regulation. A third type exists called cooperativity. The binding of one substrate to an active site will stabilize all of the other active sites. So this does not bind to the allosteric, but to the active, but it functions a lot like the allosteric activators that we see. Another control mechanism for enzymes is feedback inhibition. This is a metabolic reaction where you're trying to basically reach an efficiency. So if you have an enzyme that's biosynthetic, for example, feedback inhibition typically is very, very common. In this example, we have an enzyme that produces the isoleucine amino acid. You make that from threonine. So if you have a great deal of isoleucine in your environment, say you ate a meal that was high in isoleucine, your enzymes will inactivate themselves so that you don't waste the energy product. So redox reactions are the most basic and sort of energy transferring reactions that are occurring in biological organisms. This is two reactions that always occur together. So if something is oxidized, something is reduced. And it really is the movement of electrons. So since sodium is giving up an electron and chlorine is accepting the electron, the sodium is becoming oxidized and the chlorine is becoming reduced. So if you look at the example of, say, methane, for example, if the methane is giving up its electrons to the oxygen, that oxygen is going to turn into water as it's reduced, and that is going to be a combination redox reaction. So cell respiration and photosynthesis are also redox reactions. But oxidation is the loss of electrons, reduction is the gain of electrons. And so we can often find this in biology because when something is oxidized, the oxygen is going to go up. When something is reduced, the oxygen is going to go down. The hydrogens typically go up, and that's all because we are dealing with an aqueous situation. So oil rig is a good one. Leo Gur is another one. You lose electrons in oxidations, you gain electrons in reduction, but you do need to have an understanding of these processes. We can't just have charged particles and free radicals and these ion forms going wherever they want in the body. So over time, RNA molecules have combined to become electron carriers. We know this is RNA because there's an OH at the second carbon. This electron carrier functions like a charged battery and sort of moves material around, moves electrons around as needed. So as electrons are extracted from your food along with protons, those electrons and those protons combine with the positive form to become the charged electron carrier form. So NAD plus becomes NADH, and that is going to be the way we move electrons around in cell respiration and photosynthesis. So cell respiration, if you didn't have respiration, if you just did combustion, all of the bonds that exist within these molecules would be released at once, and that would kill any host organism. Cell respiration allows for a gradual, controlled release of energy through an electron transport chain, using oxygen as the final electron acceptor, allowing us to harness that ATP in a safe and efficient way. Overall, cell respiration has a couple of key steps. You have glycolysis, pyruvate oxidation, the citric acid cycle, and the electron transport chain. The electron transport chain and all these structures are doing phosphorylation. We are storing energy as ATP. By adding phosphate groups to ADP molecules, we make ATP, and that energy can be used all over the body in whatever cell that it's found in. So we're going to look at a closer step at each of these as we go forward. Glycolysis is the most ancestral form of energy capture. Glycolysis, sugar splitting, you are applying a small amount of ATP, 2 molecules for every 1 glucose, in order to destabilize the glucose molecule and cause it to split. When it splits and it lyses, 4 ATP are released, as well as 2 electron carriers of NADH, and those molecules are going to be used for their energy. What are we left with? Well, through the process, you're left with pyruvate. Pyruvate is a 3-carbon sugar molecule, and that's where most of the energy of the sugar still is. The pyruvate will then be carried into the cell using protein channels and a variety of active and passive transport systems, and then it will be oxidized. You'll remove a CO2, and that will leave you with an acetyl sugar, a 2-carbon sugar. That oxidation step allows us to harness a small amount of NADH, and then the acetyl group is combined with a coenzyme, which is just a helper molecule that will aid in the next step of the reaction. The Krebs cycle, or the citric acid cycle, will then break the remaining sugar bonds. The sugar is released as CO2, and in the process, you're able to extract NADH, ATP, and FADH2. The acetyl group is where the sugar is, or where the energy is. And through the reaction, the acetyl group is broken down through a variety of redox steps, allowing for that energy capture. So the sugar molecule at the end of the citric acid cycle is gone. The citric acid cycle does not, however, make much in the way of ATP, and even what it does have, some of that energy is used to regenerate the cycle, to make it a loop and bring it back around again. What's left, though, is a great deal of electron carriers from all of the previous reactions, and those electron carriers are what will move forward. So the electron carriers are brought into the electron transport chain. The electron transport chain exists at a variety of free energy steps, and it's like a stair step. Each step is going to be going to lower and lower and lower free energy. So it's going to be going from the least electronegative to the most electronegative. So oxygen is the most electronegative molecule that we're using in this process, so it becomes the final electron acceptor. As those electrons move through a series of redox reactions between every one of these protein molecules, the electrons are harnessed to create energy, or to build energy. I suppose create's a bad word. But those protons are then pumped in each of these complexes as the electrons flow downwards. And that creates a proton gradient, and that is our goal. The electrons are going to bind with protons and oxygen to make water, and that will be a waste product, But what we have is a proton gradient that is built up in the cristae between the inner and the outer membrane of the mitochondria. That proton gradient will power ATP synthase through a process called chemiosmosis. This is a proton motive force. So it's not a charged process. If we had to spend ATP to make ATP, it wouldn't be efficient. But the movement of the protons through this structure, that creates that proton motive force that gives just enough energy to bind an ATP to an ADP. And that allows for the buildup of ATP and the complexity of life as we know it. So if you break it down overall, the substrate level phosphorylation, where you're actually breaking the chemical bonds of the sugar, occurs in glycolysis and the citric acid cycle. So you're able to make a total of about 4 ATP in those steps. If you then take all of those electron carriers to oxidative phosphorylation, you can make significantly more ATP, and that gives you this total number of ATP that you see here. Now this is if you were to break down every bit of the glucose, and that's not actually likely. Those sub pieces of glucose are used to build all sorts of things throughout the body. Some of those protons are used for movement and chemical reactions and cell signaling. So this is a maximum, not an actual realistic amount. We'd have to do some efficiency calculations to find the actual amount for any particular type of cell or type of organism. So it's going to be different from, say, a stomach cell and a brain cell and a heart cell. They're going to produce very different amounts. Outside of cell respiration, we have a couple of connected processes. One of these is fermentation. So there are many, many different types of fermentation. You are expected to know too. But fermentation allows for the rebuilding of NAD+, so that glycolysis can run even when oxygen, that final electron acceptor, is not present. Humans, we don't do alcohol fermentation. Things like yeast and bacteria do. But in that process, glucose is turned into pyruvate and then to acetaldehyde and then into ethanol or alcohol. That alcohol is eventually going to build up and kill the cell. So that's why yeast, for example, are able to build to about 12% alcohol before they can't live anymore. So it's not the ethanol that's the goal here, it's the NAD+. Humans and a variety of other bacteria use lactic acid fermentation. Notice that this stays a 3-carbon sugar from glucose through pyruvate into lactate. You are able to get the NAD+ back to keep glycolysis running, but that 3-carbon lactate can be reversed back to pyruvate by spending a little bit of energy in, say, the liver to get that pyruvate back. So there is a loss of efficiency, but lactic acid fermentation is significantly more energy efficient as you can reverse the process and harness some of that energy rather than waste. Now, fermentation is different from anaerobic respiration. Some organisms use anaerobic respiration instead of cellular respiration, and while there are many functionally different steps, the main idea is that they're using something other than oxygen as the final electron acceptor. So it would be incorrect to call fermentation and anaerobic respiration the same thing, because there is no respiration in fermentation. They are not the same at all. So overall, we've discussed cell respiration as a catabolic process, the breakdown of materials, proteins and carbohydrates and fats and nucleic acids too. They are broken down into their subcomponents, and they are then turned into pyruvates through glycolysis, and acetyl groups, and then the citric acid cycle breaks down the remainder, and then all of that is carried to oxidative phosphorylation. Different types of molecules will be catabolized in different ways. So fats, for example, the alcohols will go into glycolysis, where the fatty acids will go directly into the acetyl form. So they're not all the same. Proteins, you'll notice NH3, that amino group, those are waste products. They are discarded, and that is where the nitrogenous waste comes from for things like urea. So this catabolism is just one half of the picture, though. If you actually look at all components, we'll find that the catabolic processes are linked to anabolic processes. So the breakdown of things leads to the buildup of things and vice versa. All of these things are interconnected through the combination of anabolic and catabolic reactions. The coupling of these mechanisms allows for the efficiency of life. Now, photosynthesis is fundamentally different. This is taking light energy, photon energy, and harnessing it to fix carbon molecules, and those carbon molecules become the basis of the carbon skeletons of all biological macromolecules. So photoautotrophs are organisms that are capable of doing photosynthesis. Chemoautotrophs use chemical forms instead of light energy, so they're going to use redox energy. But all sorts of organisms in the ocean and on land, they have evolved ways to do this. Bacteria did it first. Cyanobacteria still produce about half of the world's oxygen, and then plants collectively produce roughly the other half. How does this happen? Well, you are trying to capture that photon energy, and that's where the chloroplast really shows its merit. The chloroplast has a great deal of surface area in the thylakoid. There's tons of pigments that are going to be light acceptors within that structure. It's going to take CO2 in, it's going to produce sugar, and it's going to create a waste product of oxygen, and those are going to be connected. Now, plants don't do this for us. They do it for their own growth because they have both mitochondria and chloroplasts, so the oxygen released will go to their mitochondria first, but they produce more oxygen than they need, which is good because we like to breathe. Photosynthesis overall is still a redox reaction. The CO2 is becoming reduced into a sugar, storing that energy, and the water is becoming oxidized into O2. This is the best example I know of oxidation because you're literally producing oxygen. All of the atoms that you see, they're built up from the same process. Of course, the reactions are needed, but the light reactions are going to be the first major step. Light is going to be absorbed along with water. The NADP+, which is the major electron carrier here, is going to be coming as an input with the ADP, and then that is going to be producing ATP and NADPH. So the light reactions take in water, it uses the protons and the electrons to make energy, and oxygen is released as a waste product. The energy is gone from the light reactions. The energy released from the light reactions will power the carbon reactions, the Calvin cycle reactions. CO2 will come in along with those reduced molecules of energy, and the Calvin cycle will fix that inorganic gaseous CO2 into a solid sugar, CH2O for each one, or G3P for the normal product release, and it takes 2 G3Ps to make a glucose. In order for photosynthesis to be possible, there must be something to absorb the light, and that's what pigments are. So pigments are just chemical molecules that absorb light at different wavelengths. So chlorophyll A, chlorophyll B, the carotenoids, the anthocyanins, the xanthophylls, they all absorb different wavelengths of light. And what they absorb is shown in their absorption spectrum. Whatever they don't absorb is reflected, so plants look green because chlorophyll doesn't absorb a lot of green. The action spectrum is different from the absorption spectrum because the action spectrum actually measures the total rate of photosynthesis by measuring O2 release. So you'll notice that the bands don't exactly match because the action spectrum is the sum total of all pigment activity. Chlorophyll, of course, is the main one. Chlorophyll is a modified structure. You could see the structure here with the porphyrin ring and the hydrocarbon tail. The hydrocarbon tail allows it to grip into the cell membranes or the thylakoid membranes. And different pigments of chlorophyll are going to have different functional groups. But that chlorophyll is able to absorb light energy. It takes the electrons of the chlorophyll to an excited state. And when the electrons fall back down, heat is released as well as energy. So if you don't have that chlorophyll linked into an electron transport chain of some kind or protein structures, the energy will just be released as fluorescence. And that's what's going on here with this pink material. This is chlorophyll, a green chlorophyll, releasing the energy that it had absorbed because it's just a pure chlorophyll extract. So pigments harvest light. You have lots of different pigments in these photosystem structures, and those photosystem structures will pick up photons and will pass them to a special pair of chlorophyll A molecules in the center of the photosystem. Those electrons will be excited and then will be passed through the proteins of the electron transport chain in the thylakoid membrane, just like we saw in the electron transport chain of mitochondria. So every step of that train is going to be releasing energy. So that light is absorbed, it's passed to the P680 of photosystem 2, where it then moves to its primary acceptor, and then it falls down the steps. The electrons have to be replaced in the chlorophyll, and we're going to get those electrons by breaking a water molecule, and that's called hydrolysis. The electrons, as they move down, will power the cytochrome complex, which is a fancy name for a proton pump. Those protons will be pumped into the thylakoid for later use to make ATP. Those electrons will then go to photosystem 1, where it passes to P700. Those P molecules are just special chlorophylls, and 680 and 700 are the wavelengths that they absorb the best. The electrons are re-excited, and then they fall down again to power the NADP reductase enzyme to produce the NADPH. Now, we don't need as much NADPH as we do ATP, so every now and then the ferredoxin, the FD, will redirect the electrons back to the pump to be used up, and that will allow us to produce more ATP. So basically the analogy here is like this. So you apply energy to make it up to a higher level, and then gravity pulls it down, except it's electronegativity, not gravity. And those are the major flows of electrons. Those proton pumps are going to be building up protons in the thylakoid space, in the lumen of the thylakoid. You're also going to get protons from that hydrolysis split of water, and all of those will combine into a proton gradient that will power ATP synthase. So the pH is going to go down, that difference creates potential energy, the proton motive force will lead to ATP production. So ATP and NADPH are the final products of the light reactions, and both of them will go to power the Calvin cycle in the next stage. The Calvin cycle, or the Calvin-Vinson cycle, is where you are going to take the inorganic carbon from the air and you're going to fix it into a solid organic form. So fixation is completed by the enzyme Rubisco, which combines the CO2 with the 5-carbon RUBP, hence the RUB part of Rubisco. That will then produce an unstable 3-carbon sugar. We'll apply ATP and NADPH to reduce that unstable sugar into a stable form, G3P. That second major stage is called reduction because you're using the reducing power of ATP and NADPH. One G3P will be released in order to produce glucose and all of the other organic compounds, where the remaining G3P will be used to regenerate RUBP by applying a little bit more ATP and allowing this to be a cycle to continue again. Really, each enzyme of Rubisco is only able to fix one CO2 molecule at a time, but 3 Rubisco molecules will be working together to produce a stable G3P. Now, photosynthesis has evolved from aquatic systems through terrestrial systems, and one of the biggest limiters is oxygen and water loss. So plants on land have these open stomatas that allow oxygen in, or out, I should say, and CO2 in, but also water is going to be evaporating out. Now, we absolutely need that water to evaporate out. Evapotranspiration is essential for plant production, but if they're losing too much water, then the stomata will close. The cells will become flaccid and slap together. Okay, Rubisco is a very, very old enzyme. It evolved before O2 was common, and it cannot tell the difference between O2 and CO2. So if Rubisco fixes O2, it's going to go through a photorespiration process and break sugars down, rather than the Calvin cycle processes that are building sugars up. And this is very bad for plant energetics. So we have seen that there have been some major evolutionary steps to deal with this problem. In C4 plants, they use a spatial separation, so the CO2 is fixed to an organic acid by PEP carboxylase. That CO2 is then released in the bundle sheath cells, a separate cell, for Calvin cycle to occur. So the oxygen from the light reactions will build up in the mesophyll rather than the bundle sheath, and the Calvin cycle Rubisco process is not going to be harmed. In the CAM, the crucillation acid metabolism processes, CO2 will be taken in by PEP carboxylase and stored as an organic acid during the nighttime, so that the Calvin cycle will have plenty of CO2 during the day, so that even though O2 begins to build up through the light reactions, the CO2 will significantly outmeasure it. So it's a temporal separation of steps rather than a spatial one, working together to allow these efficiencies. Because you have to make these extra steps, is it going to cost some energy? Yes, but it's significantly less waste than photorespiration would have accounted for. Prokaryotes are connected to this idea because prokaryotes are the base of most of the food chains and decomposition processes. So really you just need to have a general idea that prokaryotes show all of the energy capture possible. So they are photoautotrophic, they are chemoautotrophic, they are photoheterotrophic, they are chemoheterotrophic. All of these things exist. Now it might be the black smokers of the deep ocean, or it might be these Thermus aquaticus bacteria in the hot springs, but energy is absolutely essential for all organisms, and prokaryotes are not alone in this. Plants have a lot of energy effects. So desert plants, for example, xerophytes, they have many different ways to increase the efficiency, to increase their energy capture, to decrease their water loss. Something like the Octio plants are only going to have leaves when water is available. They're going to do lots and lots of photosynthesis in a very, very short period of time. And then once the drought comes, or once it's desert again, the leaves will fall off, the flowers will fall off, and all of the energy will be stored for the next major water cycle. Oleander has crypts near their stomata that allows for humidity to build up in the crypt and allows for less water to be lost. The old man cactus has these white modified leaf spine structures that reflect a great deal of heat, decreasing the water loss. Epiphytes are going to be growing on plants. They're not parasites, but that allows them to get energy from higher sources to store water. Orchids are a great example of this, and the staghorn, which is one of my personal favorites, you can see on the left. Another option, of course, is to go parasitic. Mistletoe is a photosynthetic parasite, so it still produces its own sugar, but it gets water and nutrients from the tree that it grows on. Dodder is a non-photosynthetic parasite, so it gets everything from the plant that it grows on. And then the Indian pipe is actually a non-photosynthetic parasite of the mycorrhizae, the symbiotic fungi that help plants grow. And so all of these things will be ways of harnessing that plant energy from something else, for something else, parasitic. And then another major way is obviously carnivorous plants. They don't actually eat the meat. They kill the bugs to increase the fertility of the soil around them. Venus flytraps, pitcher plants, sundews are all great examples of this. So these plants are able to capture living organisms and modify their own soil, which allows for a great deal of life to flourish in places where the soil is not good. Okay, obviously, we also have energy shaping of evolution of animals. The shape of your teeth can give you a lot of information about the type of food your ancestors ate. The more flat teeth, the more herbivorous, the sharper tearing teeth of the carnivore, or something like humans, where we have both because we are omnivorous. The stomach of a cow is another great example of symbiosis and evolutionary interactions. They have multiple stomachs full of bacteria that are able to break down the cellulose. So the cow feeds on the grass to feed the bacteria, and then the cow feeds off the bacteria. Bunnies, they can't digest cellulose very well, so they are caprophagers. They will eat their own waste products so that the more times it goes through, the more energy they're extracting per cycle. Carnivores are much likely to have a long small intestine and a very short long intestine. Why? Well, there's not a lot of moisture there, so they need a lot of time to break down those complex proteins, which happens mostly in the small intestine, but they don't need much in the way of a cecum because they don't have massive colonies of bacteria digesting cellulose, and then the colon is very short because they don't have to extract a lot of water. Whereas something like a koala is going to have a fairly short small intestine because the protein digestion and all that is really unneeded, but a lot of the energy is redirected to the cecum, where the bacteria that is breaking down the cellulose will live, and then the colon is very, very large. The large intestine is very long because there's a lot of moisture that has to be absorbed before they release their waste products.